Hello 1001. This video is just to give you some feedback on the material from Unit 3.1. So we had a chance to look over your What Did You Learn assignments. We summarized them and kind of identified some major themes that were confusing for a lot of you. And so I'm going to try to address them here in the short little video. But of course, if you have questions that aren't addressed here, please feel free to reach out to me or Tom. We're happy to answer those questions directly. But I'm just going to go over some key things. One of the big things that was confusing for 3.1 was the cell cycle. So I'm just going to switch to slides now to walk you through the cell cycle. So we have here the outline of the cell cycle with key phases identified, and the cell cycle is really describing generally the growth process of cells in our body. So the big chunk of time cells for the big chunk of time, cells are in this called interphase, which runs from G1, S, and G2. This is when the cell is basically going through the process of generating or duplicating its genome. It does that through the process of DNA replication, and it's getting ready for this phase, mitosis, the phase in which it kind of goes through the process of dividing up into two daughter cells. The actual division of those cells is called cytokinesis, but that the steps that are involved in that division process are happen in mitosis. So while the cell is duplicating its genome, it might be doing a lot of other things, like, depending on what type of cell it is in your body. If it's a liver cell, maybe it's also generating proteins in the process of digestion and breaking down certain chemicals. Whatever the cell happens to be doing, in addition to getting ready to go through mitosis. So we distinguish between interphase, mitosis, and cytokinesis, the actual splitting of the cell. Most of the time it's in interphase. These check marks are indicating points at which the cell has internal mechanisms to determine whether or not it's actually healthy and it can proceed with growth. You don't want an unhealthy cell to grow, obviously, because an unhealthy cell will just perpetuate unhealthy cells. It will just create copies of itself that are unhealthy. In fact, that is what the basic disease of cancer is. It's when cells have acquired mutations that prevent them from adequately checking if they are healthy and perpetuating either out of control or in a way that does harm to your overall health. So there are proteins that check whether or not DNA is damaged. If it's damaged, does it need to be repaired? There are proteins that check, or enzymes that check whether or not the chromosomes have replicated properly, because each daughter cell is going to have the exact same number of chromosomes with the exact same set of homologous chromosomes. There's proteins that check are the chromosomes aligned properly. That if they're not aligned properly, obviously you're going to get two daughter cells with different combinations of chromosomes, and that's not what you want. You want to maintain genetic identity, or you want to maintain the same genetic identity. And then finally, are the growth factors present to signal cell division? So this is checking to make sure that the cell is dividing at the right time. You don't want cells to divide out of control. Other things indicate when the cell should die. Some cells in your body go through a natural life cycle and then die so that they can get replaced by new, healthier, more functioning, or better functioning cells. In fact, a lot of the cells in your body are replaced on a continuous basis. Some cells, like blood cells, your entire set of blood cells is replaced in the span of 120 days because they get beat up so much by passing through tiny capillaries. Other cells, like your bone cells, are replaced on a scale of 8 to 10 years. So every 10 years, you have a complete new skeleton. And it's all through this process of cell growth. So the next thing I wanted to talk about was just making sure you understand the difference between sister chromatids and homologous chromosomes. Here we're looking at a single cell that has one pair of homologous chromosomes. What that means is that they're the same, but they might have different copies of a given gene. Because as a diploid organism, you got one chromosome from your dad and one chromosome from your mom. And so you got the same genes on those chromosomes, but they might have different alleles. At the beginning of mitosis, these chromosomes have replicated. They've undergone DNA replication, and now you have two identical copies of each of those chromosomes. those identical copies are called sister chromatids. And so we call these set of sister chromatids a homologous chromosome pair, right? You have one set of sister chromatids here, another set here. These are both considered homologous chromosomes during that process of mitosis. And of course, this protein complex, the center of the chromosome, is the centromere. So where in this process of the cell cycle can mutations occur that lead to cancer? Well, one of the big ones is in these genes called proto-oncogenes. And these genes are responsible for triggering or signaling to the cell that it's okay to start dividing. It's okay to start growing. That's their job. Normally, these proto-oncogenes function in a healthy manner. They indicate and tell the cell to divide at the correct time. But if there's a mutation in these genes, what can end up happening is cells will start to replicate out of control and start basically gumming up the system, right? They start producing more cells than are needed, and maybe those cells then start to acquire mutations and they suck your resources and they have severe health consequences for you. This is an example of tumor growth, massive tumor growth resulting from gene mutations in the proto-oncogenes, okay? Those are telling the cell to divide at the start of G1. So that's happening at the G1 phase. The other type of genes that we see resulting in cancer when there's particular mutations are these tumor suppressor genes. And these are involved in identifying DNA repair. So a normal functioning tumor suppressor gene says, whoa, hold on, before we continue with the cell cycle, I found an error in the genome. Let's fix it. If there's a mutation in that error-sensing gene, in these tumor-suppressing genes, then they're unable to fix those mutations and they accumulate and they can lead to unregulated cell growth, or they can lead to the production of proteins that don't function correctly, which can have their own particular health consequences. And so those are the two broad flavors of mutations that can arise that cause cancer. Things that prevent the cell from regulating its own growth are things that prevent the cell from fixing damaged DNA. Another question a lot of you had was related to where these mutations happen and how that relates to inheritance. So mutations that occur in the germline are mutations that can be inherited because the germline are the cells that go through meiosis and produce gametes. And of course, gametes are the cells that pass your genetic information on to the next generation. So anything that happens in those can be transferred to the next generation. Somatic mutations are mutations that happen in cells, in basically any cell that isn't a germline cell. So it can happen in a liver cell, a lung cell, an intestinal cell, a brain cell. It doesn't matter. Because those cells aren't responsible for passing on genetic information from one generation to the next. Those mutations will only affect your health. They will not affect the health of the next generation, at least from a genetic standpoint. So that's the big difference in terms of an inherited mutation and a non-inherited mutation. Of course, if you inherit a mutation from your parents, you can pass that mutation on to the next generation, because that mutation is in all of your cells. It was in your initial embryo, and it proliferated through the process of cell division into every subsequent cell, including your germ cells. So that's the big difference. I hope that helped clarify some things. Of course, if you have any follow-up questions, please feel free to reach out. Otherwise, have a good one.